Welcome to Manifested Publishers.
Welcome learners, my name is Stephen Kariungi. Today we continue with our topic of discussion, and the topic is cell physiology. In our previous lesson, we discussed one physiological process, and that is diffusion. So today we are going to discuss the second physiological process, and this is osmosis. Now, osmosis is one of the physiological processes, or is one of the processes by which materials get in and out of the cell. Now, when you talk of osmosis in relation to diffusion, we can say that osmosis is a special type of diffusion that involves water molecules. So basically, we are saying that osmosis involves water molecules. Now, how do these water molecules move? These water molecules move from where there is high water concentration to where there is low water concentration across a semi-permeable membrane. So you can say that this is the movement of water molecules specifically from a region of high water concentration to a region of low water concentration across a semi-permeable membrane. So in that definition of osmosis, there are a few things that we need to capture. One, it involves the movement of water molecules. That's one. Then these water molecules move from a region of high water concentration to a region of low water concentration. And then this happens across a semi-permeable membrane. Now, to explain this further, we can say that a semi-permeable membrane is one that only allows small molecules to pass through but not large molecules. Not large molecules. So it is also called selectively or partially permeable. It's called selectively or partially permeable. It means the same thing as semi-permeable membrane. Now, we're going to have an experiment to demonstrate osmosis. Using a whisking tubing. And before we look at that experiment, it is important to understand what is a whisking tubing. A whisking tubing is a commercial is a commercial synthetic tube that plays the role of a semi-permeable membrane. Is a tube that works the same way as the cell membrane of the cell. So the whisking tubing, just like the cell membrane of a cell, is semi-permeable. So you can say that is a commercial, it's bought, synthetic membrane. In fact, is transparent. That acts as a semi-permeable membrane during experiments. So that being the case, we are going to have a setup so this is at the beginning. So we have here distilled water. Then inside the whisking tubing, we have sugar solution. So this is the whisking tubing. And of course, it is tied at both ends. So we have that. So that is water. That is distilled water. So on this, how you set up the experiment is that we get a whisking tubing. You tie at one end. Then you put some sugar solution up to a certain level, and then we tie the other end to make sure that the sugar solution does not spill out. Then we take that whisking tubing and immerse it into a beaker. This is a beaker that contains distilled water. So we can hang it using a string. We can hang the whisking tubing inside the distilled water using a string, and then we leave it like that for about 30 minutes. We leave the setup for about 30 minutes. And this is what we shall observe after 30 minutes. So you'll find that the whisking tubing swells or increases in volume. That is the same string we had used. We have distilled water. The sugar solution is still inside. And of course, this is the whisking tubing. Okay, you've labeled that. So this is the observation that we make that is after 30 minutes. After 30 minutes. So we are saying that the whisking tubing swells, increases in volume. The distilled water is still outside, but the volume of the sugar solution increases. That's what is making the whisking tubing to swell. Now, let's look at the explanation for that. Now, the explanation for that is that in the beaker, that is inside the beaker, there is a high concentration of water molecules. In fact, there is a higher concentration of water molecules than inside the whisking tubing. Tubing. So, as a result of that, water molecules move by osmosis from the beaker to into the whisking tubing. So we are saying that water molecules move from the beaker into the whisking tubing by osmosis. So that tells you that the water molecules have moved from where there is a high concentration of water to where there is a low concentration of water inside the whisking tubing. So now, so that is an experiment that we can carry out to demonstrate osmosis, showing that water molecules move from where there is a high concentration of water molecules to where there is a low concentration of water molecules. The sugar solution has a low concentration of water molecules. So the molecules of water will move from the beaker into the whisking tubing by osmosis. The whisking tubing in this case is the semi-permeable membrane. So you can say here that the whisking tubing is the semi-permeable membrane, and it represents the cell membrane. It represents the cell membrane of an organism. It represents the cell membrane of an organism. So basically, that's about osmosis. We can also define osmosis in a different way using the same experiment. We can say that osmosis can also be defined as the movement of water molecules. It has to be water molecules from a dilute solution, to a concentrated solution through a semi-permeable membrane. So basically, we have seen that the water molecules moved from where the distilled water, this one is the dilute, to where the solution is concentrated. So that's another definition that we can use to define osmosis, but we have to always remember that it takes place through the semi-permeable membrane. So we are going to have an assignment on that. So the first question, osmosis is a special type of diffusion involving water molecules. Explain that statement. Number 2, describe an experiment to demonstrate osmosis. And then 3, what does a whisking tubing represent in a cell? So we'll stop there. Until next time, goodbye.